Welcome to the Health Headliner podcast by Tudu Digital. Our content is informational only and not medical advice. Please consult your provider. Now, let's dive into today's headlines.
We often hear about these big infectious disease threats hitting the headlines, but sometimes it's the ones that have been sort of simmering quietly. They're the ones that suddenly explode.
Exactly.
And that seems to be what's happening with a virus called Oropouche across Latin America, the Caribbean. Since late 2023, the reported cases just skyrocketed. We're talking over 20,000.
Huge jump.
It really is.
Yeah.
I mean, to get a sense of how dramatic that is, think about this. In past decades, most countries would maybe see, a few cases a year?
If that.
Right, just a handful. It wasn't really on the radar for many.
But what really made me stop and, pay attention were these reports. 2 young, apparently healthy women died. And serious pregnancy complications linked to it. It really makes you wonder, what is going on? Is this some kind of new threat?
Well, that's the key question we really need to tackle today. Oropouche virus isn't actually new. It was identified decades ago.
But this recent, massive surge in cases, it absolutely demands we take a closer look. So we're going to delve into just how widespread this virus actually is, how it spreads and what the science is telling us about its potential to spread even further.
And we have some solid science to base this on, right? This isn't just speculation.
Not at all. Our exploration today is deeply rooted in a pretty significant new study. It was just published in the Lancet Infectious Diseases. And we'll also pull in some other related research to help paint the full picture of this evolving situation.
So where did this main study get its information? What kind of research are we talking about here? It sounds like quite a bit of data.
Yeah, it's comprehensive. We're mainly drawing from 2 places. First, there's a health news article reporting on this big study, which gives us a good overview, kind of the public health angle.
Okay.
But more importantly, we're going deep into the scientific paper itself. This paper lays out the findings from analyzing, get this, over 9,400 blood samples.
Oh, that's a lot.
It is. And what's really valuable here is that these weren't just collected recently because of the outbreak. They were gathered between 2001 and 2022.
Oh, okay. So historical data.
Exactly. Across 6 different countries in Latin America: Bolivia, Brazil, Colombia, Costa Rica, Ecuador, and Peru. So that historical perspective is it's essential for understanding why we might be seeing such a dramatic increase now.
Right. So, okay, our goal for this deep dive then is to figure out why this virus, which maybe seemed rare before, is suddenly causing so much concern. We'll look at what this science reveals about its actual spread and, really importantly, what it means for everyone listening. It feels like this is more than just another scary headline.
I think so. It's about uncovering some perhaps surprising truths hidden in the data.
Okay, let's jump into the study then. What was its main purpose? What were they trying to find out with all this work?
The goal was really threefold. First, they wanted to figure out the actual prevalence. How many people across Latin America had actually been exposed to Oropouche virus in the past?
Okay, the real baseline.
Exactly. Second, identify factors that might put someone at higher risk of getting infected. What makes transmission more likely?
Makes sense.
And third, using all that data, they hoped to predict which geographical areas might be most vulnerable to future outbreaks. Where should we be watching closely?
Testing that many samples over 2 decades. That sounds like a massive job. How did they actually do it? What was the method?
It was definitely a huge undertaking,
Yeah.
and they used a multi-pronged approach. The main technique was what's called a serological survey.
Serological, so blood serum.
Correct. They tested those 9,400 plus anonymized blood serum samples looking for specific proteins, antibodies that our bodies make in response to virus.
Right.
They focused on 2 types: immunoglobulin G, usually called IgG, and immunoglobulin M, or IgM.
And they tell you different things, right?
They do. IgM usually pops up early in an infection. IgG develops a bit later, but can stick around for a much longer time, indicating a past infection. They use standard lab tests, commercial ones called enzyme-linked immunosorbent assays, or ELISAs, designed specifically to find Oropouche antibodies.
So if someone's blood had that IgG antibody, it suggests they had the virus at some point, maybe even years ago, even if they never got diagnosed.
That's exactly the idea. And, to be really sure, These tests were picking up Oropouche specifically, and not antibodies to some other related virus.
Because viruses can be similar sometimes.
They can be, especially within the same family. So they did extra testing on a subset 71 samples that were positive for IgG. They used a more specific technique called plaque reduction neutralization testing.
Okay, what does that do?
It basically checks if the antibodies in the blood can actively stop the virus from infecting cells in a lab dish. It confirms specificity. They tested against another related virus, guarovirus, and also 2 Oropouche cousins, you could say, Iquitos virus and Madre de Dios virus. These are called glycoprotein reassortants.
So really drilling down to make sure it was Oropouche.
Precisely. And they didn't just use one version of Oropouche. They tested against 3 different strains. One from the recent Cuba outbreak, a 2020 Peruvian one, and an older historical strain from Brazil.
That's some serious validation.
Yeah.
Okay, so they figured out who likely had it in the past. How did they then try to connect that to risk factors and predict future spread?
Right, that's where the spatial modeling comes in. They used pretty sophisticated machine learning techniques. One is called Lancet, or maximum entropy.
Machine learning. How does that work here?
Well, they feed the model all the information about who had antibodies and where they were located. Then they layer in tons of other data, environmental stuff like climate, temperature, rainfall, land cover.
Like forests versus cities.
Exactly. And demographic data, too, like altitude, how close people live to towns or cities. They even included data on where the insects that carry the virus are known to live.
The vectors.
The vectors, yes. So they built 2 main models. One used the antibody data to understand patterns of past infection. The other used the actual reported case numbers from the recent 2024 surge to see what might be driving the current situation.
Fascinating how they can combine all those different layers. The outline also mentioned genomic analysis. What was that about?
Yes, another key part. They gathered all the available genetic sequences for the Oropouche virus that researchers have shared publicly over the years.
The virus's genetic code.
Right. And they used this to build phylogenetic trees, basically family trees for the virus, showing how different strains are related and how they've evolved. They also looked for signs of what's called selection pressure. Meaning, are there specific genetic changes that seem to be helping the virus survive or spread better? And they did lab experiments, too, comparing how well different Oropouche strains grew in cell cultures, trying to see if the recent outbreak strain had any, unusual growth advantages.
Okay, wow. A really thorough investigation from all sides. So after all that work, what did they find? Let's start with the big one. How common is this virus based on those antibody tests?
Yeah, the antibody results, the seroprevalence, were quite eye-opening. On average, across all 6 countries, 6.3% of the samples had those IgG antibodies.
6.3%. That sounds significant.
It is. It suggests a noticeable chunk of the population in those areas has encountered this virus before. But, and this is important, that average hides some really big regional differences.
Like what?
Well, for instance, in Costa Rica, it was less than 2%. But you go to the Amazon region, and it jumps to over 10%.
Over 10% in the Amazon. That's way higher than the few cases usually reported, isn't it?
Massively higher. It really points towards past circulation that wasn't picked up. They also found a link with altitude people living higher up were less likely to have antibodies. And as you might expect, the rate increased with age, suggesting ongoing exposure over time.
Okay, that Amazon figure is striking. What about those spatial risk models? Where did they predict problems could arise?
The model, based on the historical antibody data, strongly pointed to the Amazon River Basin as the highest risk area, which fits perfectly with the 10% finding there.
Makes sense.
But it also highlighted coastal and southern Brazil, parts of Central America and the Caribbean as potentially higher risk zones. And what was really compelling was that these predicted high-risk areas lined up pretty closely with where the big 2023-2024 outbreak actually happened.
So the model had predictive power. What factors seemed most important in those models?
Climate. Overwhelmingly, climatic conditions were the biggest drivers, things like rainfall patterns, steady temperatures, overall precipitation. These factors accounted for over 60% of the variable contribution in the model.
60% just from climate.
Yes. It really underscores how much the environment influences this. And the researchers specifically suspect that the recent El Niño weather phenomenon likely created favorable conditions that, well, fueled this latest surge.
So it's not just about the bugs, it's about the weather that helps the bugs, or maybe the virus itself.
It seems to be a complex interplay, yeah. The right conditions can boost vector populations or perhaps even affect virus replication or transmission efficiency.
Okay. Now, about the virus itself, did they find any smoking gun, any changes in the virus that explain this huge jump in cases?
That's one of the most intriguing parts. Surprisingly, no. When they compared the genetic sequences and the lab growth characteristics of the 2023-2024 outbreak strain against older ones,
Oh,
they didn't find significant differences in how it interacts with the immune system, its antigenicity, or how well it grew in the lab. No obvious signs it had become inherently nastier or more transmissible.
huh. So the virus hasn't changed much.
Exactly. It points back to those antibody numbers. The study strongly suggests that Oropouche virus infections have been massively underdiagnosed and underreported across Latin America for years, maybe decades.
So the virus was there all along, just flying under the radar.
That seems to be the most likely explanation based on this data. The high antibody rates compared to the historically low reported case numbers. It's a huge discrepancy. Many infections were likely mild or misdiagnosed, perhaps as dengue or other fevers, and just never confirmed as Oropouche.
Okay, that shifts the perspective quite a bit. So what are the big implications here? Why should people listening really pay attention to this?
Well, several big things. From a public health standpoint, knowing the virus is this widespread that many people have been infected silently, it changes the game. The recent surge, plus those concerning reports of severe cases and potential pregnancy issues.
Right, the deaths and complications.
Yes. It highlights an urgent need for, first, just more awareness among doctors and the public, and second, much better, more accessible diagnostic tests. We need to be able to identify it properly.
And the climate connection you mentioned, what are the implications there?
That's potentially huge. The strong link between climate factors and virus spread suggests that as our global climate patterns continue to change,
Which they are.
which they definitely are, the geographic range of Oropouche could expand. Outbreaks might become more frequent or occur in new areas. So understanding these environmental drivers is absolutely critical for predicting risk and preparing for the future.
So for people living in these areas, or maybe thinking of traveling there, what does this mean practically? How should we protect ourselves?
It really ramps up the importance of standard insect bite prevention, consistent use of long sleeves, long pants, using effective repellents. Look for ones with DTA or icaridin.
Okay.
And here's a key point about Oropouche. It's mainly spread by tiny biting midges. You might know them as no-see-ums.
Oh, those annoying little things.
Exactly, and they are tiny, just a few millimeters. The problem is they can often get through the mesh of standard mosquito nets.
Really? So a regular bed net might not be enough.
It might not offer full protection against midges, so in high-risk areas, using finer mesh netting is probably a good idea for better protection, especially while sleeping.
Good tip. Now, the study mentioned potential harm during pregnancy. What's the advice there?
Yes, this is obviously a serious concern. The researchers noted reports linking Oropouche to miscarriage and potentially congenital issues, while they suggest it might be less frequent than with Zika.
Which caused huge problems.
Yes, but any risk is concerning. So the advice right now, especially while infection rates are high in certain regions, is for pregnant women or those planning pregnancy to consult their doctor before traveling to known Oropouche hotspots. It's about informed caution.
It sounds like this study answered a lot, but also opened up new questions. What are the big research gaps now? Where do scientists need to focus next?
Absolutely. This study really highlights how much we still don't know. We need much more research on the long-term health effects of Oropouche. Does it cause chronic issues? We don't know. And definitely a clearer picture of the impact on pregnancy and babies. That's crucial. Beyond that, more detailed studies on the specific midge vectors, which species are most important, where do they live, how do we control them?
Vector control, right.
Yes. And of course, the classic needs development of effective vaccines and better, faster, cheaper diagnostic tools that can be used widely.
It sounds like a very solid study, but like all research, I assume it had some limitations we should bear in mind.
Oh, definitely. The researchers are usually upfront about these. One point is that the highly specific neutralization testing, the PRNT,
The one that confirms the antibody target.
right, they couldn't do that on all 9,000 plus samples, only on that subset of 71. It's resource intensive. So there's a small possibility some of the initial ELISA positives could have been due to cross-reactivity with other viruses, though the overall patterns seem robust.
Okay, what else?
Another unknown is exactly how long those IgG antibodies stick around in the blood after an Oropouche infection. Is it years? decades,
Yeah.
lifetime, we don't know for sure. And that could slightly affect how we interpret the percentage of people showing past infection.
Right. If they fade quickly, the real number could be even higher.
Potentially, yes. Or if they last forever, it reflects cumulative exposure over many years.
Any limitations with the spatial modeling part? Predicting risk areas.
Yeah, a couple of things there. The accuracy depends heavily on the data you feed it. They noted limitations in, for example, having complete continent-wide climate data for the very recent period, like all of 2024, when doing the modeling.
Data gaps.
Exactly. Also, for technical reasons, they couldn't incorporate age into the spatial models, which might have added another layer of understanding. And finally, using existing sample collections always carries a small risk of bias. Maybe the people in those collections aren't perfectly representative of the general population. Though they did check this and felt the type of collection didn't hugely skew the antibody detection rate.
Good to know the nuances. Now, Professor Jan Felix Drexler was a key researcher here. What were his main takeaways, his sort of expert opinion on this?
Professor Drexler really hammered home that finding of massive underdiagnosis. He stressed that as a critical point. He also strongly emphasized the role of climate, pointing to things like El Niño as likely drivers for outbreaks. And looking ahead, he voiced concern that climate change could, unfortunately, lead to the virus spreading even further.
So a warning there.
Yes, and his practical advice echoed what we discussed. Prevent insect bites rigorously, and pregnant women really need to consult their doctors about travel to risk areas.
And how did the study authors collectively sum up their findings in the paper itself? What was their final interpretation?
Their main conclusion reinforces that climate is a major driver, potentially amplified by extreme weather like we saw in 2024. They also highlighted that while the main Oropouche strains seem antigenically similar, those related reassortant viruses, like eith, might interact differently with our immune system, something needing more research.
Okay.
But the bottom line was clear. Better preparedness needs better diagnostics, stronger surveillance to track the virus in real time, and smarter vector control strategies, guided by the kind of risk mapping their study demonstrated.
This has been incredibly informative, really shedding light on something that maybe wasn't on many people's radar. If you had to boil it down, what's the main message or the call to action for our listeners?
I think the core message is awareness and precaution. If you live in or are planning travel to Latin America or the Caribbean especially, those areas highlighted, like the Amazon, coastal Brazil, parts of Central America understand that Oropouche is likely far more common than official numbers used to suggest.
So take the risk seriously.
Yes. Take insect bite prevention very seriously. Remember those tiny midges and maybe think about finer mesh nets. Stay updated on local public health advisories. And crucially, if you have concerns, especially if you're pregnant, talk to your doctor, get personalized advice, don't hesitate.
That's really clear, practical advice. And, thinking beyond just Oropouche, it really makes you consider the bigger picture, doesn't it?
Yeah,
this whole landscape of potentially underdiagnosed tropical diseases.
Absolutely.
And how large-scale environmental changes, like climate change, might be fundamentally reshaping infectious disease threats globally. It kind of leaves you wondering, what other hidden viruses might be out there, more prevalent than we think? And what can we actually do as individuals, as societies, to get ahead of the curve, to better understand and prepare for these emerging health challenges? It's definitely food for thought.
Thanks for tuning in to the Health Headliner podcast by To Digital. Like and subscribe for more health news and breakthroughs. Stay healthy and informed. See you next time.